RL1s program with the NCBCs. There have been more than 100 different RL1s reviewed under that, and more than 30 different RL1s collaborated, and you'll see them during these talks. Okay, so you just heard a special session on a presentation on imaging as relates to that, and the way we're doing it is there are 3 half-hour presentations that are aggregating different flavors, kind of cross-cutting a transposition of the different domain sciences. So the current one that we're going to build up to now is systems biology and computational science, and Russ Altman will give the presentation, and then the last half an hour, Mark Mewson will give a talk on clinical informatics. So Russ, take it away.
Great, thank you. Is this working? Good. So thanks very much, Peter, for the introduction. And this has been a very exciting program starting in 2004, as Peter mentioned. My task today is to describe the results of 3 centers, and it's not going to be fair and even because I'm involved with the third one, and I'm using the slides that have been communicated to me by my colleagues from Magnet and NCIBI. But the theme will come through in all 3 slide sets. That I think the underlying theme of this session is: was this program worth doing? And when you kind of pick a 1 or 2 sites, or 8 sites in this case, to be national centers, do they do things that are more than just good research? Because good research can be funded with investigator-initiated grants, or with program project grants with 3 or 4 investigators. And I really, and building upon what Ron just said, I think there's very clear evidence that these centers, because of being called centers and because of the way it was expected for them to behave, 2 things in addition to excellent science came out. One was a forming community, and I think Ron just gave you a great idea of this medical imaging community that has crystallized around the NAMIC Center and its sister and brother centers. So there's community that happens, and that leads to more rapid improvement of the infrastructure in the field, because people talk to each other and realize that they're doing a lot of the same things, that there are best practices. The second thing that happens is the hardening and distribution of really high-quality software that many of us are not motivated to do if we just are doing science. In fact, at Symbios, and I'm quite sure it's true at other centers, the people who are often the first authors on the science papers are not the primary architects of the hardened software that is delivered to the community for reuse. These are different people, they have different skill sets, but by bringing them together under the National Center umbrella, they work with each other, they know each other. So that really is my bottom line, and you'll see in these 3 centers that has happened, and I'll try to point that out 3 times in case there's any doubt that I really believe it. So Magnet is the multi-scale analysis of genomic and cellular network center. It's led by Andre Califano at Columbia. His colleague, Aris Floratis, is here, and so can answer detailed questions. But the way I think about Magnet, and since I'm talking, I guess I'm the one who gets to characterize it, this is a very excellent systems biology group that has been not only doing high-quality systems biology, mostly of cancer, but has been leaving along the way valuable multi-purpose tools that can help other people do the same exact analyses in their favorite systems. So what the approach has been is a very, I think, exemplary systems biology approach, where genomic data, epigenomic data, certainly transcriptomic data is gathered in collaboration with experimentalists, and then non-trivial but very innovative analytic resources are created to understand the cell regulatory logic. And of course, cell regulation is key for cancer, and so cancer is a very natural application area for these tools, which then yield insight into the control of cancer and yield targets that can be potentially used to treat cancer. So Magnet is centered in Columbia, and I guess this is some kind of map of Columbia and New York and Manhattan. It's a very exciting place. in their overall strategy now I'm going to go a little bit under the covers for what these grants look like, because I think there was a certain genius in the design of these programs. Everybody is supposed to have a core computational research effort, and then driving biological problems that drive that computational tool building with real-world constraints from real-world scientists. So in the case of Magnet, we can see that the core is atomic level molecular interactions. Let me just point at these for a moment. These are the computational cores. Models for genetic variability and models for understanding regulatory interactions. They build, they publish, and they harden software to do this. Then they test the software on the driving biological projects, which are shown here on the outside. One of them relates to the gene family Hox and its specificity of its biological interactions. One focuses on the ERB signaling cascade, and one works on prostate cancer interactions. And you can see there's a nice little buildup from molecular to cellular signaling to tissue and disease. Very typical organization and I think really one of the good ideas that the framers, if you will, of the NCBC program introduced. So this is just what I just said. These are a little bit more about the leadership of the project. One is at the atomic level, led by Barry Honig and colleagues. One is led by Andrea himself, looking at regulatory molecular interactions. There's one on genetic variability. And then there's the software engineering. And this is also very typical. These are people who harden the software that the students and the postdocs who are perhaps not trained in software hardening develop. And then, as I said, there's the DBPs, driving biological problems. This idea has percolated into lots of other grant mechanisms at NIH. The idea that all of us computationalists, computational bioinformatics people, should be driven by biological problems that not necessarily rule our life, but keep our algorithmic work relevant is not a bad idea. And it's very consistent with the NIH mission. So as I said, the role of DNA shape in understanding the Hox family, probabilistic dynamic models of ERB signaling, and the discovery of master regulators of tumorigenesis in prostate cancer. Again, Andrea provided me with this slide, which is a great and I think typical slide that shows the value of these national centers beyond the publications. So Magnet has done a great job publishing. They've had collaborations. They could show a map very similar to what Ron just showed of a national and really international map with little dots on the people who are either collaborating directly with the center, who have written R01s that collaborate and use the tools of the center, or who are friends of the center doing other kind of collaborative activities. They create infrastructure both for computing and for software. Without exception, I believe, the national centers have had educational programs in which workshops at conferences, outreach sessions like the one we're having here are created. And then, of course, probably the most important outcome of the national centers is the human product. The students, the postdocs, who then go on to have careers is incredibly important. And what these national centers provide that might not otherwise be provided is an early exposure to the importance of a sense of having a field and an exposure to the scientific resources that can be available when you collaborate. So I think that the students and postdocs that are coming out of these programs have an expectation for a national infrastructure that we never had growing up. As I was a young academic, I had no idea how I would interact with others, where my computing infrastructure would come from, and now there's a partial answer. I will, if I might make an editorial comment, these will be sunsetting after 10 years in 2013, 2014. I think it's an absolute mandate for the country to come up with the next phase. What are we going to do next to make sure that the momentum and the precedent set by these centers is continued? Because it would be terrible to lose this huge investment of both software, hardware, and personnel talent. Andrea and his colleagues have published, based on the driving biological problems, in great journals. We congratulate them. I'm not going to go over these, but I think this, again, is not atypical of the centers where there's scientific output that backs up the utility of the tools. And then, of course, there's the other things that they do, and software dissemination. I should say that the GE Workbench is the vehicle through which many of the tools developed at Magnet get put out to the world, and they've also have a very successful conference. So I'm not going to repeat all that for the NCIBI. I'll tell you that this is the National Center for Integrative Biomedical Informatics, led by Brian Athey and his colleagues at the University of Michigan. And they have a focus on metabolomics and genomics and creating tools for the integration of those data sets. So again, I think the thing to highlight here is the expression data, the genetic data, the metabolomic data, bringing it together and then integrating it to create both scientific results and tools that are available to others. They have adopted a web service model. So instead of downloading their software, you interact with it over the web. They have created several tools in this area, which I won't go over. And then they've gotten involved in the same kinds of outreach activities that you heard earlier from Ron and in my description of Magnet. One of the interesting things that they're doing that I wanted to highlight is this Transmart public-private partnership for open source clinical translational informatics tools. I think many of you know that translational bioinformatics is of great interest these days. The connection to the clinic, absolutely critical. Many of us who spent much of our lives looking at 3D structure or genomic data are now being introduced to the electronic medical record, medical images. This is incredibly exciting, but there are special purpose software that is needed. You heard about I2B2, which has had a lot of penetration. You'll hear probably a little bit more about it from, I would guess, from Mark. And this Transmart effort fits right into that as kind of a social structure to bring people together for translational bioinformatics. That's all I'm going to say about that. I think I used all the slides that they sent me, and I'm sure that Brian would be happy to answer questions about their mission. And so now I just want to give you a rough idea of what is happening at our center, SymBios, the National Center for Physics-Based Simulation of Biological Structure, which I really co-lead with Scott Delp and Vijay Pandey at Stanford. The two scientific foci at SymBios have been biomechanical simulation at the scale of a human and molecular dynamics simulation at the scale of molecules. And then we've had smaller projects that are trying to bridge these length scales. Those are very much developmental. That's a hard thing, and I'm not going to discuss this. I'm going to discuss these two briefly because we have spent a lot of time with very high quality engineered software that's available for download today in both of these areas. So first, OpenSim and the biomechanics. So OpenSim is an open source simulation environment for biomechanics. What do I mean by biomechanics? Well, there's somebody walking. I'm going to go through these very quickly, but one of the things you can do with OpenSim is a simulation of a person running. Media not found. Okay, imagine, if you will, a spectacular image of this skeleton running across the screen. While he would be running, I don't know if it's a he. At this level of representation, it's quite irrelevant. You would see the muscles represented as these blue lines lighting up in red when they were activated. And so Scott Delp will be very irritated with me. It was working this morning. But this is the kind of output that we produce from OpenSim. You can then look at such models to understand the forces on an ankle when it's injured. This is an example of the OpenSim application. It's a very simple cycle, really. You build a 3D model, you type in the parameters, then you hook that up with an engine that applies Newton's rules of physics to integrate the forces over time, and you recompute, and then that gives you your dynamics, which you can then study either graphically or either with a picture of a person running, or more typically in the papers, you'll see these graphs of the key parameters of interest. OpenSim allows the biomechanical community, and I'll talk about the uptake, which has been spectacular, to build models of high-quality models of particular biomechanical units, like a knee or a spine or an entire running model of the lower. Notice there's no muscles at the top, so this is just focusing on the lower part, or the shoulder. They're all in a standard format. They can be uploaded into a resource, and then they can be shared. This is the kind of acceleration of progress I was talking that the NCBCs can affect. Scott's group would never have spent the time to build and harden open source code just on an R01. You need that extra resource and the community, and I think this is a theme you heard from Ron, and I'm sure you'll hear from the other speakers. So then you can put this stuff on the web, and you can share and accelerate. These are literally web pages on our site called simtk.org that not only present the model, but the full documentation, the community of people who use that model. It's all there, and you can enter the field and get up to speed much more quickly than 10 years ago. Of course, OpenSim is a resource. There's a community forming around it. For every national center, you can see a map like this. It tends to be focused in the United States, but in many cases goes global immediately. I'm quite sure that's true for all of the national centers. Of course, then we do science with this. We look at accuracy. We look at how well we understand the kinematics of various human motions. People use OpenSim to understand the effects of heavy backpacks on, in this case, a soldier, in terms of their gait, the strain on their muscles, and how to support them and allow them to go further and longer with 50 pounds of material. You can make metabolic assessments by linking the biomechanics to calculations of work and power that are done by the simulated people. You can look at injury risk. This is all work done outside of SymBios by members of the community using OpenSim. You can scale a shoulder that you built for a child to an adult. Those are usually nonlinear scalings, and so they're not trivial, but then you can get a benefit from a model built in one context and apply it to another context. Like all centers, we get involved in training. The OpenSim program has had several workshops, often with conferences like this, where people can take an extra half day to, in a hands-on environment, build a model so that they get to see the software working. And I think it's fair to say that the next generation of biomechanical modelers are being raised up using OpenSim. In the latest work, we're actually hooking OpenSim to a neurally instrumented monkey so that we can read and when I say we, I mean my colleagues at SymBios, so that we can read the impulses in the motor cortex for the monkey to move its arms, and instead of and intercept those signals and send them to an OpenSim model of an arm. This is very early work for amputees or other people having artificial limbs controlled by their motor cortex. Very early, obviously. Okay, now I just want to give you a very brief 2 minutes on the other scale where we've made a significant a significant effort, and this is in the area of molecular dynamics simulation. This is a G-protein coupled receptor, which is a membrane-bound receptor whose family is responsible for 30 to 50% of all drugs. This is what that looks like in a membrane, where you have lipids around it and water at the extracellular and intracellular side. Part of SymBios' mission has been to build fast and powerful simulation techniques that allow this system to be simulated on time scales that are relevant for drug response, which is milliseconds. Because of the physics, we have to take time steps of femtoseconds. To get to milliseconds is a lot of time steps. And so my colleague Vijay Pandey has been leading up a team that has innovated in this area. And one of the solutions that Vijay introduced before SymBios was getting everybody to donate their screensavers to the task of simulation. And that got a certain amount of speed up. Then part of the earliest contributions of SymBios was to move molecular dynamics onto these unbelievably powerful graphics cards that can do 100 to 1,000x faster floating point operations than many CPUs. With that, we were able to achieve something like 100-fold speed up. And then we combined these ideas and got people who bought Sony PlayStations to donate their extra cycles to folding of proteins. As of a couple of years ago, 10% of the machines on the network that were devoted to protein folding were PlayStations, but they were donating more than 90% of the CPU power. That's how powerful these NVIDIA and other cards are for computation. It's an incredible bear to program them, but once you program them, they're incredibly powerful. And then most recently, this is not enough, by the way, to get to microsecond scales or millisecond. So then we've been looking at Markov modeling of the state transitions that you see during the simulations, and Vijay and colleagues and the community have now created tools that get us another 10 to 1,000-fold speed up by doing simulations smarter. You don't just turn on the simulation and go get a cup of coffee. Well, you still can go get a cup of coffee. But what the computer is doing is it's watching the simulations, and when it notices that it's exploring spaces that have already been explored, it shuts down the simulation and branches it off into an area of structure that has not been explored. That's, of course, a simplification, but those kinds of ideas give you 100 to 1,000-fold more efficiency. So let me just give you, so OpenMM is available today. You can download and you can use it. If you have one of our supported video cards in your laptop, you can do 100x or 1,000x faster simulations now with OpenMM. It's an LPGL software license. It's open source. And what we've done in the last couple of years is focus on embedding OpenMM into the most commonly used molecular structure packages, AMBER, CHARM, and others. People like those user interfaces, but using OpenMM, we can make the back ends much faster. And we're working with all of those developers to do that. We've also written a Python interface, so you can even, and we love this. This is very nerdy, but this is a 12-line Python program that builds a molecular dynamics simulator. And that's just, I think, amazing, and I won't say anything more. So one of the things we can do with this is we can do simulations that are biologically important. This is a long simulation, so I'm not going to show the whole thing, where we see the spontaneous formation of a micelle. Where does it say micelle? A micelle is a bunch of lipids where all of their hydrophobic tails get together in a center, and their hydrophilic heads orient towards the outside. And in a simulation, we just added these in solution, and they spontaneously form a micelle, which you can kind of see the green hydrophobes starting to assemble in the center here, and the blue and orange hydrophilic groups on the outside. This is a cross-section of the simulation. I'm not going to go over that, but I can say that because of this increase in capabilities, simulations have gone from 36 amino acids, de novo folding, from an extended chain to a folded structure, and now we're pushing 100 amino acids and things that really begin to look like proteins. This is kind of a map of where we are in terms of time and where we are in terms of capabilities. Was Symbios responsible for all of this? Of course not. But we were part of an ecosystem that kind of crystallized with the creation of the national centers and then is moving forward together. And I think that's the story that's told 8 times by the national centers. I think I'll almost finish up to show you one of the kind of very satisfying, this is an extended chain of a small protein called villain. We're not showing the water, but there's a full water and salt environment. There's also 2001 Space Odyssey playing on my laptop, but you can't hear it. The important point is this is a de novo fold of a small peptide. And this was about 36. We're now doing this for 100. Maybe you can hear it. Okay. I'm going to skip over this because I know that there's not time. I will also say that in the most recent years, we've been looking at protein-ligand interactions, such as for drug. And here we have a small, this molecule L6, interacting over 2 microseconds with its target. And this is a totally unbiased simulation where we put them in the same box, turn on the physics, and then watch it kind of snuggle into its binding pocket. The idea now would be, can we do this routinely for drugs? Can we design new drugs? Can we use physics to understand drug action? Okay. There's also work in Alzheimer's disease, which I won't discuss, but tell you obviously it's very exciting to be able to use these tools, partner with clinicians, and start coming up with hypotheses about new drugs or new treatments. Okay. Underlying all this is the simtk.org site, where people can put their software, their data, and their models. It is like a source forge, but it's biologically oriented. So you could use SourceForge, but then you might get lost in a sea of other things, or GitHub. But in SimTK, there's a community that comes that knows it's biological. We have 21,000 registered users on SimTK. We have something like 600 projects that are making available their code. So I would encourage you, in my lab, before you can graduate or finish your postdoc, you must upload all your code and documentation onto SimTK. So certainly all outputs of the Altman lab for the last 6-7 years are on simtk.org. I would encourage you to do this, because as you know, when a student leaves, it sometimes is very stressful to know where's their code and how does it work. And so if you make it a standard practice that they should post and document, it makes passing the project on to the next student or postdoc infinitely easier. I'll finish up by saying we publish a magazine called the Biomedical Computation Review. It comes out on the web, but also in hard copy. We'd be happy to send you hard copy. If you go to the website, you can just put in your address. And then I will stop with my conclusions, which are that the three centers focus on science, community building, and infrastructure. The community building and infrastructure does not come for free, and that's why the National Center idea has legs, in my opinion. And the key of success is producing and integrating all three of these, and in the case that I just showed you, in the context of systems biology and computational biology. So I'll stop there, and thank you.
One short question, and then there's a 1 hour, half hour at the end where you can interact more completely while we do the switch over.
Can I ask one short question?
Yes.
So Russ, justify, given the brutal funding environment that we're facing,
Yes.
why we need to integrate. Why can't we fund science and R01s and then fund infrastructure and community building at somewhere less than $4 million a year times 2 years?
Because infrastructure and community building are expensive and important, and they're as important.
Why not have to be integrated? Why
Otherwise, the community building and the infrastructure, in the same way that a bioinformatician needs to be driven by biology, those community building and infrastructure activities must be driven by the relevant science. So if they're not people sitting together in the same center, understanding what's today's biggest problem, what does the community need to know about, which infrastructure is worth making incredibly expensive. So obviously these are professional software engineers, so they make a lot of money. So you do not want to spend a cent, you don't want to waste a moment of their time. And so by having them sitting shoulder to shoulder with people who are active in science, you guarantee a pertinent and relevant community and infrastructure. And I've come to believe that if you have these activities be separate, you will be wasting all of your infrastructure and community money because they will not do the most pertinent things. And that's what we could debate, but I think that that's what the experience of the 8 centers demonstrates. It's the key issue for the National Center program.
I'm coming here as a chair of the special session. We need to
Would you like to?
Okay, I'll do it this way. Hello, everybody. I know that it's the break of 5 minutes of moving, but really on the behalf of ISCB, ISMB, I really want to thank the organizer. There is a lot of work on organizing this special session. So to Peter and to Jen, thank you very much. This is a small sign of appreciation from the society, so thank you very much.